I added another, even more recent review article that directly refers to some of the IPSC stuff. I think we left off here, maybe somewhere along in there. And now everybody knows you have to memorize every detail of every protein, and you have to look up the function of every one of these proteins. Oh no, as I say, you won't be tested on these details. But with anything, I feel like you have to understand a little bit about the normal function, what the protein is supposed to be doing in order to understand, what's happening with the disease. Okay. So the only thing you need to know about this is find where dystrophin is. Okay, so this is the outer membrane of muscle cell, intracellular, extracellular. Okay, that's all on the outside. Don't worry about any of these things. All I just want you to appreciate is that there's this massive protein, dystrophin, that is responsible down here. It connects to the actin cytoskeleton. What's the importance of the actin cytoskeleton? Or what could you imagine a component of something called the cytoskeleton might be important for? Yeah, structural integrity of the cell, right? And that I would say that's particularly important because muscle is contracting, and all muscles contract. Skeletal muscles, I didn't look up, I don't remember the details of the difference between skeletal, cardiac, and smooth muscle, but there are differences. Yet they're still muscle, and they're still contracting. Okay. So, dystrophin plays a really important role in that because it's attached here inside the cell to the cytoskeleton, but then it's involved in both mediating the structural aspects of all of these proteins that interact with the extracellular, as well as there's some chemical things going on here having to do with NOx, nitrous oxide synthase. That biochemistry people, anybody know what nitrous oxide synthase does, what nitrous oxide is? Anyway, it's got, okay, and then the other thing to get from this diagram is that there's specific domains of this massive protein that are doing different functions. Now, the interesting thing, it's still kind of striking about, unusual, curious about this protein, as we'll see, you can actually restore quite a bit of normal function, even if there's huge chunks, internal chunks of this protein that are missing. Not 100% function, but enough that it holds a lot of promise for therapy. Okay. Not that it's going to completely alleviate the symptoms, but as we discussed the other day, right now there's absolutely no treatment for Duchenne's muscular dystrophy. Those boys that can be, diagnosed at the age of 3 or 4 or 5 are going to be confined to the wheelchair for the rest of their life and having various other problems, and eventually are going to develop cardiac problems, and eventually they're going to die by the time they're in their 20s. No treatment. Okay, so anything that would, even alleviate a little bit of that symptomology would be fantastic for both those young men and their families. Now, I touched on this the other day. Is there, so what's the inheritance pattern of Duchenne's and Becker's muscular dystrophy? I've only been talking about boys. Can females have it? Can females have it? It's an X-linked recessive, so absolutely no females would have any symptoms, anything like muscular dystrophy, correct? You could, it's a lot less likely, but I do have a slide in here where there are some explanations where there are women that have those symptoms. There is one case that I read where a young girl was actually homozygous for X-linked recessive muscular dystrophy. That is possible, right? But then wait a minute, most of those boys don't survive until they're in their 20s. Well, the father had a relatively mild form, the child, and then the mom was a carrier, of course, and the child just unfortunately was homozygous and she was pretty severely affected. But there's some other ways that women that are not homozygous can have symptoms. They're not the full-blown Duchenne's, but yes.
Is it because the carriers have, they're like symptomatic carriers?
Yeah, there's a term for that. Symptomatic carrier is one of those terms. What's another term? I've got it in the slides. Maybe I didn't have it.
Is it the other kind of, it's not the other kind of muscular dystrophy, is it?
No,
Okay.
no. Symptomatic carrier and manifesting carrier. They're synonymously used. But this is a woman who is heterozygous for the X-linked recessive disorder. Yet she's showing symptoms. How could that be? She's got, yes,
X inactivation?
X inactivation. Yeah, we're used to thinking, and you might have been taught that it should be random. You would think that in any given cell in an XX individual, 50%, one cell would have a 50% chance of having the maternal X inactivated, 50% chance of having the paternal X inactivated. Well, there is what's called skewing, where there's, can even be as high as 70% inactivation of one allele, and if there's 70% skewing, and if the 30% of the X chromosomes are the ones that are carrying the wild type allele, and you've got 70% inactivation of that wild type allele, you're going to have some symptoms. And there's other reasons, other ways too. Okay, so let's talk a little bit more about the protein. It's huge. I don't think it's anymore, it's the actual, anybody know, actual largest gene and largest protein known. There's a cardiac heart protein called tintin or something like that. I think might be bigger, but this is, if not the biggest, it's one of the very biggest. Look at this. Anybody ever done Western blots? What size of the proteins are you looking for on a typical Western blot, or what size were you looking for?
Like 500, or not 500.
Western? Philadelphia?
Yeah.
Yeah, 500 is pretty big. I don't think that's, anyway, this is big. The average is less than 400. So maybe you were working on a particularly large. Anybody else working on a large protein? Anyway, that causes technical problems with doing jelly with fruit for Western blots for people, and people struggled with this for quite a while, but then they did get it. And the other thing too is that almost 4,000 amino acids. Okay, 2.4 megabases, but then a lot of that, of course, is intron. There's a lot of splicing. It takes 16 hours to transcribe this gene to the precursor RNA, and then of course that gets, I don't know how long for a complete. Did I say translate? I wrote translate. I think that's transcribe. I don't think it's 16 to translate. Ribosomes are a lot faster. I think that's a typo. Sorry about that. But anyway, it gets processed down to 14 kb, 2.4 megabases to 14 kb in a cDNA. 14,000 nucleotides. Okay, lots of exons. Now you don't have to know all the details of all of the color coding and all the different domains, but this does. This is from that extra supplemental review article that I assigned. So here's the actin binding domain. Okay, these spectrum repeats, we'll come back to them. Not so much the details of them, but just where a lot of the deletions are taking place here that cause both Duchenne and Becker muscular dystrophy. These little hinges, they all have to do with the 3D structure. And then just this cysteine-rich region at the end, don't worry too much about this, but this is the C-terminus. So this is on the outside, the actin binding N-terminal is on the inside. Now, what you do need to pay attention to, especially to understand the Youngerthal paper and how they were going about making deletions, big deletions, that restored function. The original allele that we'll talk about, are alleles that Young et al. Used, caused severe Duchenne's muscular dystrophy. They had big deletions. What they intentionally did was use CRISPR to make even bigger deletions to restore function. Now, how can that happen? Well, it's because if you understand what's going on here, see how each of these is a exon, whether it's a nice rectangle, square, or whether it's got these. But now pay attention to the shape of each of these. If they're splicing this 4 to 5, 3 to 4 to 5, and this one being, not having the triangle shape at the end, these are all in the same reading frame. Is that reading frame for transcription or reading frame for translation? Reading frame for translation. Okay. So now this is the normal mature mRNA, at least for, at least what's functioning in skeletal and cardiac muscle. But if you have a mutation or a deletion, and you ended up, let's look at, if you splice, instead of splicing from exon 7 to exon 8, if you splice from 7 to even 9, if you just skip exon 8, look at the shape of these two exons. Even though 7 and 8 are in the same reading frame when they're spliced together, and 8 and 9 are in the same reading frame, if you skip 8 and splice together 7 and 9, you're going to get a different reading frame. What's going to happen to the rest of the protein? To the C-terminal side of that altered reading frame? Everything is different. Is it just going to keep on translating all this and then terminate at the normal translation termination codon? Why are you saying no? Yeah, so where is the termination codon going to be?
Either earlier or after.
Can it be earlier?
I mean, you could earlier.
Oh, earlier than this
Yeah,
one. Yeah, okay, I thought you meant
Oh, yeah,
earlier,
no.
before, yeah. So everything downstream to that out of frame splice is gonna be out of frame, and just somewhere in that sequence, there's gonna be a termination codon that will now be in frame with the altered reading frame. It would be, it's not a nonsense mutation. That's a misconception that some people will have. If there's a different reading frame, and that encounters the normal sequence, no change in the sequence at that codon, but in that reading frame, it's a termination codon, that's what the ribosome doesn't know. That's not the normal termination codon for the reading frame it's supposed to be used, it just terminates. And just anywhere in the sequence could be, that termination. Now, one thing that you don't have to know any details about for this paper, and I'm not going to test you on it, but one thing that you might have heard of, have anybody heard of a mechanism where the cell can kind of compensate for premature termination codons or even nonsense mutations? Is there any kind of surveillance mechanism for that you've ever heard of? The nonsense surveillance mechanism, nonsense mediated decay. So a lot of times those mutations that cause nonsense mutations and even frame shifts that cause premature termination codon, that will signal the nonsense mediated decay, and that normal message will actually increase its breakdown. Okay, now that's a problem, right? If that's the only allele, but hopefully there would be some other, even if they're minor splice products that can help out. Okay. So that's the problem. Now pay attention, so understanding this, the shape of the exons and understanding which of them, which splicing events, whether it's in the wild type or because of a mutation, like we can have mutations in splice sites that would cause aberrant splicing, exon skipping. We can have deletions. The breakpoints of the deletions might be entirely within introns. But what they can do is then cause splicing, but then once they're spliced together, the downstream exons will be out of frame. And that's the problem, but that's also what gives both gene therapy, which we're not going to talk about a lot, but also gene correction, a lot of potential for ameliorating at least some of the symptoms of Duchenne's. Okay. Now, again, you don't have to know all the details of this. I've been referring to Duchenne's and Becker, just make sure everybody knows they are mutations in the same gene. It's just that Duchenne's has a much more severe phenotype. This is the one that most people know about, and it is more common. Becker's is much milder, but they both can be caused by a variety of different mutations, and it's obviously affecting the same gene. Now, this is an interesting thing you might not be aware of. What's it mean to have a de novo mutation? And is this possible that only 2/3 of the cases have carrier mothers? How can that be? How can you have say a boy born with Duchenne's and not have a carrier mother? And clearly his dad didn't have it. Yes.
So de novo is kind of like a new mutation, like unprecedented kind of in a person.
I wouldn't quite say unprecedented. Anybody want to add anything to that? Yeah.
Maybe it's possible there's a lot of sites that the mutation can disrupt the activity of protein. That's why the mutation is more likely to be you have a phenotype.
Yeah, you're still not quite getting there. Do you want to add more?
Well, I was gonna say that de novo can come from a variety, like mutations in general can come from a variety of factors, not just like inheritance. Like you can ingest mutagens or like your environment can affect your genetics.
Okay, well, let's say we rule those out.
Okay.
I mean, those aren't gonna be, yes, they
Sure.
can happen, but they're gonna be,
There
that
are
when
also
it's
mistakes.
but where are they happening is the thing.
Okay.
The mom and dad, obviously, if this is a boy, right, that got Duchenne's, he's not gonna have gotten the X chromosome from his dad, right? So we can rule out that any mutations coming from his dad, right? Because assuming normal karyotype, right? He got a Y chromosome, it had to come from his dad. So the X chromosome had to come from his mom. But let's say you sequence a bunch of the dystrophin genes in a whole bunch of somatic cells of the mom, and you don't find anything wrong with the dystrophin gene.
It's a germline.
Yes, that's what I'm getting at. Other than a few rare, very rare cases of the child having. Okay, so most of the time it is in the germline. Now there is another possibility. It could have happened a somatic mutation very early in the embryo, when that child was, 2-cell, 4-cell, something like that. Okay. But that's what's interesting about this. But then, if the kid the child, the boy doesn't live long enough, and I think that he's going to have pretty severe symptoms. I'm not sure he's going to be able to have, natural conception for the child. I suppose they could do IVF, assisted reproduction, but chances are he's not going to be able to. But the other interesting thing is that there are several cases, and this is really kind of interesting, and I want you to be aware of it. Again, there's de novo mutations, but she has more. There are several cases where the mom has more than one affected sons, like 2 of them. that's really unlucky for one thing. Mom is not a carrier. But usually we think of de novo mutations happening in the germ cells, right? And it's a rare event. And especially in the mom, go back and think about germ cell development in the females. This had to have been, if a mutation took place during DNA replication, that had to have been when the mom, way back, had been an embryo, right? So that's the only time in the female those germ cells are going through mitosis. Well, the only way to explain, and I don't know if they can prove it, a mom that's not a carrier but having more than one infected son is that the mutation actually took place early enough in her embryonic germ cells that there are multiple oocytes that have exactly the same mutation. Really unlucky, right? But it can happen. Now, the interesting thing about the genetics of Duchenne's is that there's all kinds of, this is kind of like an example where you can explain lots of, unusual types of inheritance, including the high frequency of de novo mutations. Now that's not, there are others that are just as high, but this I've never heard of before. Maybe there are other cases that they are. Non-random X inactivation for females having it, Turner syndrome makes sense, right? There's only one X, and if that X came from, the mom could have been a carrier, right? But if the daughter only has one X chromosome and that has the mutation, this is actually really interesting. X autosome translocations. What does that mean? What's a translocation? What's a translocation?
Isn't it where one section of the X chromosome and the same section on the other homolog, they like switch and
Not quite the other, don't say other homolog.
oh the other
Non-homologous.
yeah, the non-homologous chromosome.
Yes.
They like homologous sections of the chromosome
Not homologous.
not
Non-homologous. X chromosome
yeah.
and say chromosome 14,
Mm.
chromosome 10, chromosome 3. I'm not sure if there's how many different autosomes that are involved in this, but one portion of the X chromosome translocates reciprocally
Right.
with a portion of an autosome. So the genome is still balanced. There might not be any problem with a reciprocal translocation, but these were actually really, I think you can read about these in the review article, the Gerard review article. These identification of these and identifying the breakpoints were actually really instrumental in determining the initial cloning of the dystrophin gene back in the 80s. They told them it was on what portion of the chromosome was on, because what happened with these translocations is that on the X chromosome, the break of the translation actually disrupted the dystrophin gene. That's why these females had muscular dystrophy. And that's very easy to see even back in the old days of the 1980s of determining that. Okay, so yeah, this is telling you more about the carrier, the manifesting carriers. And it all gets to really interesting aspects of X inactivation. This is another aspect. Look at this. Why is this significant? Several pairs of monozygotic twins, both of them are heterozygous, and they're girls. Monozygotic twins that are discordant for genetic disorder, knowing that they're both heterozygous. What's monozygotic? Is that identical or not identical? Identical. How can one have a disease and not the other? Well, there doesn't have to be epigenetic. X inactivation was different in one twin than the other, and unfortunately the one twin that has the disease had skewed X inactivation and was unlucky. So that, again, it's another example. This is just showing you, and we'll have some more immunofluorescence. This is actually, you can't see the underlying cells that are the muscle. This is just immunofluorescence staining for an antibody against dystrophin. But just telling you, this is a normal. Okay. This is DMD. There's no protein, and this has been confirmed at the level of the Western. The vast majority of Duchenne's patients in the skeletal muscle have absolutely no detectable dystrophin protein. Okay. But you comparing the normal, and remember, dystrophin is on the inner side of the cell membrane. So basically, if you're staining for dystrophin protein, it's outlining every one of the cells in the normal. Mild, slightly reduced, mild vectors. You can have severe vectors that have even less. This is interesting. Look at this. The muscle, this whole figure is muscle. How can you explain? Now, this is a manifesting carrier of Duchenne's. So, is that a female? And is she heterozygous? Yes. But this is what clued them in on what was going on with these manifesting carriers. Some cells are completely devoid, just like the DMD, constitutive DMD. Other cells have some dystrophin protein. They have some symptoms, but they're typically not very severe. Okay, now again, this is from a really old paper, really old, 1899, just showing, kind of giving you more schematic of, these are not the exons, these are the spectrum repeats, these blue things here. Okay? But just showing you that there's these are showing you the extent of the deletions. So the arrows and arrowheads and the lines represent the deletions, but different parts tend to be more severe. And they can be mapped to different domains of the protein. Okay, I didn't want to show you any pictures of animals. I didn't want to show you any pictures of people with, trying to walk that have Duchenne's, but that's so funny.
Oh, we're talking about, because you're talking about how old that paper was.
Yeah.
And we're just talking about like what old papers would have been
Yeah, well, okay, let me just tell you that I got my PhD in 1990, so I published some of those old papers.
like.
I look back on these figures and I go, oh my God, did I publish papers like that? And I did, but that was state of the art back then. Look at some southern plots. I could go into all kinds of stories about the old days. But when I was in grad school, our professors would tell us, well, when I was in grad school, I had to purify my own restriction enzymes. I couldn't go to a company and buy them. So. Anyway, so yes, there are animal models. Now, the dog model isn't created. These are spontaneous. And there are different breeds shown here. There are, I'm not sure why. Okay, here's another thing. If you just consider on a per gene basis, you would think this gene has the highest frequency of mutations possible because there's hundreds of alleles in humans. There's lots of spontaneous mutations in domestic animals. It's not really on the nucleotide base pair basis that it has a higher mutation frequency. It's because the gene is so huge. But there's just more chance of the mutation. So there's actually golden retrievers, corgis, labs, I forgot what GSP, German short-haired pointers, and these spaniels. 4 or 5 different breeds of dogs have 5 different mutations. So it's not like there's a founder mutation in one breed and then it was bred into lots of derived breeds. These are independent mutations. The reason I'm showing you the golden retriever model is we'll come back to it in a minute. There's a whole bunch of spontaneous mouse mutations too. None of these, let's see. Yeah, none of these, all this is showing on this row, all the different spontaneous mutations in mouse. Now mouse, even with no mutations, this is the original one, don't have a severe phenotype as humans. So they're not necessarily a good model. They are to a certain extent. And people have made better models by gene targeting and making mutations in other genes and then crossing them in other genes involved in that same complex. And they get much more severe symptoms. Dogs are, in terms of comparison to the human phenotype, are a really good model in terms of the age, they start showing symptoms when they're young, but by the time a 2-year-old, well, that's a pretty old, 2 years old, you know, young adult, they're pretty bad off. Yeah. There are some other ones that have been targeted, and again, you could go to these sites if you want, and they've actually got animated videos, live videos of these pigs and rabbits. In both cases, the wild-type pig is on the left, and the knockout Duchenne's is on the right. And what you're seeing here is going from the time. The pigs they have to go up this little step, and you can see the person's hand there. They're actually holding up some food. You know, pigs like dogs, they like to go for food, right? The guy on the left, he just jumps up there and he's real happy. He's heading towards this poor little guy. He's got muscle weakness. He can't even get up the steps. And this is only a step, you know, about this high, which is sad, which is why I didn't want to show the actual live video. Same thing with bunny rabbits. They're being tempted with food. The wildlife guy on the left just hops right up and he's happy. He turns off. The poor little guy on the right isn't strong enough to be able to get up that step. Now this was made, they're both knockouts. This one was made not by CRISPR, but a targeted homologous recombination. This was made by CRISPR. So that's the end of those slides. Why don't we take a break? I'm finally getting